Okay, well, I think the number of participants climbing and joining is slowing down. So let's get started. Welcome, everyone, to the end of March Biocurator Working Group call. Today, we're excited to have the SCID-CID GCEP giving an update, and Troy will present that on behalf of the group. So we're excited to hear that. And then for the next meeting, which is Wednesday, April 9th at 4 p.m. Eastern Daylight Time, the hereditary cancer clinical domain will be giving an update on their progress. And I think that's all the announcements we have. So with that, I will turn it over to you, Troy, and thank you for coming to present.
So, thanks for the invitation. Let me get started here. Um, okay, that has to be the right one. Okay, where'd you go? There we go. Okay, so can you see? What can you see? Hold on.
I think it just needs to be in presentation mode. Okay, but now it's presenter. So if you can swap the displays.
Oh, gotcha. Okay,
Yep, looks
how's
good.
that? Okay, all right.
Yeah.
So thanks for inviting us to present. So the goals for today are to just give a brief introduction to the immune system. So again, not knowing how what everybody's background is, I thought it would be good to just kind of level set, make sure that we're on the same page, and then I'll just give you a brief introduction to inborn errors of immunity, and then we'll talk specifically about the GCEP and a little bit about the VCEP and why we chose these diseases in particular. So. In terms of when I think about the immune system, I think, trying to make it as simple as possible, I think about it in terms of these four compartments. So two of the compartments are part of what we call the innate immune system. That's the part of your immune system that you're born with, and that it doesn't really learn in the way that we think that we think about the other part of the immune system. It contains two compartments: the complement compartment, and then the myeloid compartment. These are oftentimes these are the phagocyte type cells, like neutrophils, monocytes, macrophages, dendritic cells fit in this compartment. And with as part of this compartment, the barriers that we have in our body are really important. So your mucosal barriers, your skin, they keep pathogens on one side of the fence and keep everything else on the other side of the fence, and play an important role in the innate immune response. The other half of the immune system is what's known as the adaptive immune system. And this is the part of the immune system that learns. It gets better each time it sees a pathogen or something that it's supposed to respond to, like a vaccine. And these are the B cell and the T cell compartments. And we'll be mainly focused on these today, the B and T cell compartments today. Each of these compartments, all together, they comprise your host defense, and they really interact with one another through direct cell-cell interactions and through the secretion of cytokines and chemokines, which I'll talk about in just a minute. But when I think about this, I think about this in terms of a total defense system. And so, like the defense system, say in any country that wants to defend itself, it needs to defend itself from invasion from a variety of directions. So, if you're an island and your body is sort of an island, you need to defend yourself from anything coming from the sea, from the air, from within. And each of these compartments are like that. They do a particular thing that defends you against particular types and particular the particular approach of pathogens. So I think about the complement system, which is these proteins are in the plasma and extracellular fluid. These are sort of like the like landmines. Something trips them up, and then there's a series of events, and it ultimately ends in an explosion, which is the formation of the membrane attack complex that literally blows up things like bacteria, cells, and plays and does that. Your myeloid cells are kind of like your first responders. They are often, you think about, say, the Marines that, crawl through the tissue, or that crawl, through the mud to the side of the battle. The phagocytes crawl through the tissues to the side of the battle where they may meet bacteria, viruses. They engulf those, eat them, and they then show their friends what they've eaten. And the B cells are sort of like an air force in that they make and deploy these smart bomb cruise missile like molecules called antibodies. And so they can do this whether they're right at the site of an infection or they're on the other side of the body. And lastly is the T cells that are they come in different varieties, but they're sort of the generals, the assassins, security guards, and the psychologists of the immune system. They sort of, some of the T cells are the ones that sort of tell the rest of the immune system, "Hey, settle down, don't get so riled up." So they are the ones that sort of direct a lot of immune responses in concert with these other cells. Okay, so um, when immune cells get activated, they do a few different things. One of them is that they proliferate and make more of themselves. They produce cytokines and chemokines, as I mentioned. And then another thing that they do is that immunity really happens in the tissues. For ages, we've measured mainly what's in the peripheral blood in humans, but the peripheral blood only contains about 5% of your total immune cells. So it's just part of them, and really the business part of immunology takes place in the tissues. And so what happens is as cells are exposed, say, to a pathogen, like shown here, say bacteria, one of the things that they'll do is upregulate adhesion and trafficking molecules to allow them to traffic into tissues. The other thing that they'll do is they'll, of course, get very excited. Ooh, we've got to get out there and attack this pathogen, but we don't want them to do that too much. And so they have these inbuilt systems where once they get activated, they start to put up molecules that serve as brakes to slow them down. So there's a big activation stage, as I'll show you in a second, and then the brakes get applied and start to slow the immune system down. And the reason I show you these compartments and what immune cells do when they get activated is that there are genetic immune deficiencies or genetic immune diseases that affect each one of these things. And so just to sort of put this in context, this is what we're going to be talking about. Um, so as I mentioned, that they can communicate with each other through direct cell-cell interactions, but they also do it through cytokines and through chemokines. Chemokines are what draw the immune cells into the tissues and bring them to the sites of infection. I'm going to jump over this just real quick because I think it's so. As I mentioned, the immune system, I think of it, a normal immune response, sort of like driving your car. So so there's like a green light phase, a yellow phase, a caution yellow light phase, and then a stop red light phase. So you can imagine that an immune trigger, so that might be, let's just use the example of a vaccine, for instance. Vaccines contain all kinds of foreign molecules, or molecules that are foreign to the body that they're not used to seeing all the time. You take that and you squirt it in, and it might be basically that a bacteria has been, mashed up, and that's what you're injecting. Or it might be something like the COVID vaccine, the mRNA vaccines, that you're injecting a piece of RNA that your cells take up. They transcribe that then into protein, they translate that, into protein, and then those proteins are what your immune system responds to. But it's a foreign protein, like the one, like the proteins from SARS-CoV-2. So you give the trigger. There's a green light phase where the immune system just accelerates. It's like, you're at a stop sign, light goes green, you look ahead and you say, okay, I'm going to hit my accelerator. So the immune system accelerates, and as it gets activated. And remember, I told you that as it's being activated, it's also putting up these molecules that act as brakes, because what you don't want to happen is for your immune system to just go absolutely bananas. Because if it does that, then you get into trouble, like we saw very commonly during the pandemic, when in patients who were dying because their immune systems, they were having cytokine storm. Their immune systems were going crazy, and that was actually the reason that they were dying, was because of the overaction of the immune system. That's not supposed to happen. So as the immune cells put up these brakes, those start to regulate the immune system. And so this is the red light, or this is the yellow light, the caution light. You're looking up ahead and you see the next intersection, and you see the cautions, or the yellow, the light goes yellow, and you start to slow down. You don't stop yet, but you start to just slow down and temper your speed. And then when you get to the light goes red, and you need to stop. So you hit the brakes and you stop. So in the immune system, you stop the response. But in addition to stopping the response, the immune system has to clear a bunch of stuff out that, say, from a tissue site where it had had an immune reaction. And so clearing things out might be clearing out any remains of the infection, for instance, clearing out any immune cells that had moved into that tissue to respond, and just getting rid of anything that's not supposed to be there, to bring that tissue back to a state of homeostasis. And that process is there's a resolution phase, and that process takes place here. At the end of that, your immune system is left with memories of what it has seen, and those are really mediated through the T and the B cells, as we'll talk about in a second. If any of these steps go awry, then you can wind up, if there's overactivation, underregulation, or the resolution doesn't happen, then you can wind up with autoimmune diseases, or cytokine storm, or what we call generally as immune dysregulation. And I think just it's fun to think about the fact that we ask the immune system to do this kind of impossible job, which is that we have asked the immune system to respond to any possible pathogen, anything that we could encounter out in the environment. The problem is that we also tell it, you need to respond to anything and everything. However, you can't respond to us. You can't create autoimmune disease. And so it's really quite amazing to think about this delicate balance that the immune system has, of being able to respond to virtually anything without responding to us and causing autoimmunity. It's pretty amazing that we don't all have some immune disease, really. So today, the SCID-CID GCEP. So SCID is severe combined immune deficiency. SCID is combined immune deficiency. And when we talk about combined immune deficiencies, or severe combined immune deficiencies, what we're talking about is immune deficiencies that include defects in cellular immunity. And typically when immunologists talk about cellular immunity, they're talking about the role of T cells. Okay, so when we talk about, so these are defects that have T cell problems, and then they also are combined with B cell problems. So the B cells again make the antibodies, and immunologists for eons have called anything that causes the B cell problem a humoral problem. And so there's the cellular part of the immune system, the humoral part of the immune system, or the T cell and B cell parts, and those when they're both involved in when they're both defective, then you have a combined immune deficiency. So what we're going to be, what the GCEP and the VCEP are focused on are immune diseases that generally cause problems with both of these compartments. Let me just walk you through briefly what these compartments do. So the T cells, as I mentioned, they're the generals, they're the security guard assassins, and they're the psychologist peacekeepers of the immune system. And what I mean by that is the generals are the CD4 T cells. They play an important role in directing the B cells, directing other cells what they should do. So these are often referred to as the helper T cells because they help the B cells know how they should respond to things that they see and what types of antibodies they should make. So these are kind of the generals. The assassin security guard T cells are CD8 cytotoxic T cells. And you can imagine that if one of the cells in your body becomes infected with a virus, viruses are, of course, just parasites, essentially. They take over the machinery of your cells. They then use that machinery to make more of themselves. And they start to then push out pieces of those viruses that will bud off from these cells. And that's how they make more of themselves. But under normal circumstances, our own the cells that are our own cells, they're basically, we've got old, proteins that get old and need to be degraded and gotten rid of. So they'll be degraded, but pieces of those proteins will wind up on the cell surface in these MHC. Some people call them HLA molecules. They wind up there, and they basically are a way of just telling the immune system, "Hey, I'm part of home and country. Don't bother me. Don't kill me." That's all great, fine and dandy, unless you get a virus that takes over and the machinery starts to make more of itself. Now what happens is you've got pieces of the viral proteins that get put in these MHC molecules on the surface. These CD8 T cytotoxic T cells, they see those, they say, "Hey, wait a minute, you're committing treason, you're harboring a pathogen inside you, and it would be better that you just die because we need to get that pathogen out in the open where it can be attacked by the other parts of the immune system and gotten rid of and get it cleared out." So what happens is the CD8 cytotoxic T cell, it sees that little piece of virus here in this pink MHC molecule. It binds to it through its T cell receptor, and it then snuggles up to this cell, and it begins to secrete granules onto the surface of this cell. And these granules contain a couple of important things. One of them are molecules called perforin that actually punches holes in the membrane of this target cell. And then granzymes that percolate through these holes, and they initiate programmed cell death in the target cell so that the target cell says, "Okay, you've convinced me I should die for the greater good and release this virus that's now taken me over out into the open so it can be attacked." So that's what happens. The cell dies, the virus gets released. Now any free viral particles can be eaten up by some of those phagocyte cells, or it can be bound by antibodies and neutralized. But it's a way of really rooting out intracellular pathogens like viruses, getting them out in the open where the immune system can control them. Okay, so these are the security guard assassin T cells. And then we have one more class, which are the psychologist peacekeeper T cells. These are the regulatory T cells, and their job is to basically go around the immune system and say, "Hey, we had that virus infection last week. You did a great job fighting that off, but hey, settle down, take it easy, don't get so riled up." Because if we allow ourselves to the cells in our body to stay riled up for too long, then you risk that problem of bridging over into having too much activation and risking autoimmunity. Okay, so with these things in mind, what happens if you don't have T cells? And this is the case in patients with severe combined immune deficiency. In all of the different types of severe combined immune deficiency, all have problems with T cells of some type. So they can't get, if the T cells aren't there, that you don't have the generals, so you can't help B cells to make antibody responses. And so you get recurrent infections because you can't make good antibody responses. You don't have your security guard assassins, so you can't clear out viruses very well. So intracellular pathogens like viruses, even viruses that don't bother us too much, like rhinoviruses, the classic cold virus, um, can't be dealt with. And so those become now pathogens that can be life-threatening in patients with severe combined immune deficiency, for example. And if you don't have the psychologist peacekeepers, then any cells that do get, happen to get a little bit out of control can begin to attack the host and cause symptoms that look like graft versus host disease, really caused by autoimmune processes that kick off. And because all of this is happening and you're not able to control any infections, of course, a lot of energy goes into that, and these patients often have failure to thrive and don't grow very well. So severe combined immune deficiency, these are the really worst of the patients that have these combined T and B cell problems. And this is where uh, the first part of the GCEP and VCEP focus, and I'll explain why in a couple of minutes. I'm going to jump past this. So let's jump to the B cells now. So the B cell compartment, remember that I said these combined immune deficiencies involve both the T and B cell compartment. So what do B cells do? So we've talked about the role of T cells, but B cells, their main job is to make these antibody molecules. One of their jobs is to, help detect things, but really their biggest job is to make these antibody molecules. It's the reason we vaccinate people, so that they'll make antibodies that can protect them against infections, neutralize viruses, and things like that. So what happens is that B cells go through a number of maturational stages, starting in the bone marrow and then maturing up, ultimately coming out to the spleen, usually, to sort of finish off their development. And then they're released into the body as naive B cells. And then they encounter what they're supposed to respond to, and then they mature and become more mature B cells. If you don't have B cells and you're not able to make these antibody molecules on the surface of the B cells, the most common thing that we see, if that's the only problem you have, is recurrent bacterial sinopulmonary infections. And so that means sinusitis, pneumonias, bronchitis, ear infections. So, and those are the most common problems. And so in patients with combined immune deficiencies, they have these other problems that having poor T cell function causes, so like susceptibility to viruses and things like that. But on top of that, they can't make good antibody responses. So now they can't defend themselves against bacterial insults. And for instance, they would not be able to respond to particular viruses. And so sometimes we see those viruses causing problems that way. So these combined immune deficiencies cause both sides of an inability to respond to normal pathogens. And just to walk through this, so again, if you immunize with a vaccine, it's eaten up by certain types of immune cells. Some of those myeloid immune cells eat it up. They digest it. They put a little piece of it out there. They educate any T cells that have made a response, or that are specific to this. They tell them, "Hey, you should get activated because I've just eaten something, and you should become responsive to this." That T cell gets activated. It goes over and it then talks to a B cell. The B cell has also the antibodies that are on the surface of the B cell have also seen this protein, and it said, "Hey, wait a minute, I've been put on alert that there's something here," but the B cell can only get so activated on its own. It can only go so far without needing sort of the help of a helper T cell that interacts with it and tells the B cell, hey, that thing you saw, it's real, it's out there, we need to attack it. And it's okay for you to now switch over and really begin churning out antibodies, which is what happens. It's like a secret handshake. The B cell then gets activated and it starts to churn out all kinds of antibodies. It class switches. Normal when it first the alarm goes off, it starts to make these IgM antibodies. And once it talks to a T cell and gets help from a T cell, it starts to switch over and make these IgG and other antibodies. All of the T cell receptors, the antibodies on the surface, those are how the T cells see the world through their T cell receptors. The B cells see the world through their antibody molecules. And these antibody molecules look like, I think of them as sort of, they're actually, this is a crystal structure. It's actually a Y-shaped protein. I think of them as sort of the hot dog roasters of the immune system. And what I mean by that is that there's a handle piece that the immune cells can get a hold of. And there's then the prong fork piece out on the end that you would, if this were a hot dog roaster, you'd put your hot dog or marshmallows out here. But this is where the antibody binds to, say, a virus or a bacteria out here. And there's two of them. And so, and they're exactly the same. And so it can respond, this antibody can bind to one over here and one over here. And it can create, in doing that, it can create these complexes it can cross-link pieces, multiple pieces of say, multiple viruses, bacteria can cross-link them together, and that creates sort of these big globs of immune complexes, and those immune complexes can very actively or can very strongly activate the immune system. Now, as I mentioned, the T cells see the world through the T cell receptor. The B cells see the world through their B cell receptor or their antibody molecules that are on their surface, but also get secreted. And one of the things that many of you probably know is that these are the way that these are generated is through the first process of that is a genetic process that we have in our genome on our chromosomes, these regions that encode the antibody, the genes for the antibody chains and for the T cell receptor chains. And these regions are really quite unique within the genome in that you have multiple V segments. This gene has multiple V segments, multiple D segments, and these V segments are called variable segments. These and the J segments are joining segments, and then there's these D segments that get plugged in between the V and the J segments. And basically, part of the way that your immune system creates I put you may have noticed on the title slide there was a picture of a socket set, and you may have said, what does the socket set have to do with what we're going to talk about? Well, this is how we create the socket set of our immune system, because we need to have B cells and T cells, again, that can respond to anything, really. And so how do we create that variety? Well, there's a couple of ways. One of them is that it's like having 3 bags full of different colored beads, and just as a random process, you reach in and you grab a red bead from so a V segment, and you reach into the next bag and grab a green D segment, and the next bag and grab a yellow J segment, and then you plug them together. But you can imagine if these were beads on a string, when you plug them together, there'd be sort of, so say if you chose this red segment over here and this D segment in the middle and this J segment here, when you plug those together, there'd be like these big loops of chromosome in between them. And that's exactly what happens. You've got these big loops of chromosome sticking that bleb out. And we have machinery, the RAG enzymes that are like scissors that come along, and they recognize these regions next to these specific sequences next to each of these regions, and they basically are the scissors, and they clip that off. So a piece of your chromosome gets clipped off and floats away, and the ends then get grabbed by the DNA repair machinery, and they ultimately get the ends of the chromosome now get glued back together. So now you bring that V segment and the J segment that you've chosen, for instance, right up next to each other. They get pasted back together. And at the same time, the ends of this circle piece, this loop of chromosome that got cut out, they same thing happens to them. Raw pieces, raw ends of chromosome bound by the DNA repair machinery, they get glued back together and they form what's called a T cell receptor excision circle, or a TREC. And again, the sequences right next to these segments are conserved because that's what you need to see for these enzymes to recognize that and make the cuts. Well, so somebody got the idea years ago to say, well, hey, we could quantify the number of these circles that have been made by just doing a PCR reaction between these known sequences. The sequence in the middle is variable, but these sequences on the end are conserved. So we'll just PCR between them. And if you do quantitative PCR, you can quantitate these T cell receptor excision circles, and it gives you a surrogate for the number of T cells that you have. And so this is the basis of newborn screening for SCID, in that it's newborn screening for SCID is a quantitative PCR where newborn babies should have a lot of naive T cells. Those T cells have rearranged their T cell receptors and they've just made a whole bunch of these TRECs. And so just you take the dried blood spot, you do the quantitative PCR off of the dried blood spot to quantitate the number of these TRECs that are proportionate to the number of naive T cells. And if they're there, then the patient has naive T cells. Those naive T cells are functioning. If they're not there, then you know that that patient may have really low T cells, and that's then what flags in the newborn screen to bring a patient back to clinic, because they might have one of these severe combined immunodeficiency diseases that have low T cells. Okay, so I know I went through a lot of background to get to this point, but I thought it would, I do think it's important to understand this, especially as genetic counselors, because because this is the basis for all newborn screening that's done for SCID. The other thing, and this is just for funsies, the other way that we generate diversity in our antibody genes and our T cell receptor genes. Remember, we've got those V, D, and J segments, and we randomly choose one and plug it together. But in addition to that, before those ends get plugged together, TDT actually just randomly plugs a bunch of nucleotides onto the end of the chromosome. It just like jumps into the gap. It adds randomly a bunch of nucleotides, and then it gets plugged back together by the DNA repair machinery. And so, depending on the number of nucleotides that get plugged in there, of course, they need to be in they need to be in replicates of 3 to stay in the reading frame. So anything that's not just doesn't make a productive protein, but anything that is makes a productive protein, and those are the ones that survive and make it through. So it's another way, it's a second way of creating diversity in our T cell receptor and B cell receptor or antibody genes. And just to give you an idea of the amount of diversity that we can generate with these two approaches, so it's estimated that we can generate a total potential diversity of 10 to the 14th, okay, right here. 10 to the 14th possible combinations is what we would be able to generate using these approaches. The total number of lymphocytes, T and B cells, in your whole body are estimated to be about 10 to the 12th. So you can actually generate more diversity in this process than you have total T and B cells. It's crazy, and it's pretty incredible to think about, and especially when thinking about the genetics of it is pretty remarkable. Anyway, that's just sort of for fun. Now, as I mentioned, all of these genetic SCID defects, that gene defects, all of them have deficiency of one or more T cell. Most of them have deficiency of all of the T cells. Some of them have deficiency of just part of the T cells. So that's the T cell part. And so these are the genes along some of the genes that you see over here. These are their T cell, but many of them also may have problems in the B cell compartment with the absence of B cells, as shown here in the B negative. And some of them have absence of NK cells. And it all has to do with whether the protein that's encoded by the gene that is defective is important for T cell development, B cell development, or NK cell development. And so if it's important for all of them, of course, then you wind up with a T negative, B negative, NK negative SCID. If it's important for, say, just a development of T cells and NK cells, you get a T negative, B positive because it's not important for the development of B cells, but NK negative because it's important for the development of NK cells. So this is how we sort of classify the phenotype of SCID patients as based on their T, B, and NK cell phenotypes. Okay, all right. So that's sort of the background, the immunology background. Now let me talk to you a little bit about how this fits into the global scheme of immune genes. So every couple of years, I'm on a committee part of the International Union for Immunologic Societies, that we get together and we search through the literature looking for any new gene defects that have been associated with immune diseases. And then we pull all those together and we then create a publication. And those publications are, the people in the field refer to it as the IUIS gene list publication. It's basically a catalog of all of the genes. And this is data from, in this catalog, we try to break them down. And when we started doing this now several years ago, it was relatively easy to put each of these gene defects because of their phenotype into a bucket. And those buckets were then put into tables. And so that it was organized around these 10 tables. And for instance, table 1 are the cellular and humoral immune defects. So these are the SCID defects in table 1. Table 2 is part of the CID combined immune deficiency defects are in table 1, and the ones that have syndromic features, so other syndromic features, are in table 2 of the IOS gene list table. And as I'll show you in a minute, the list is now 560 genes that have been associated with immune diseases in humans. And this is just the last time that we did this in 2022, the numbers of genes in each of these tables are shown in blue, and our update that we just barely got accepted for publication adds these additional numbers of genes in red to each of the tables. Okay, just to give you a sense, and to give you a sense for the growth of the number of genes being identified. Of course, by year, it comes down, and then of course NGS sequencing comes along, and then there's this huge explosion, which has of course been seen in other fields as well. What's interesting in these is that the classic definition of immune deficiencies, so primary immune deficiency diseases, or PIDDs, was that they had problems with infections that they couldn't clear very well. And this was the predominant cause of morbidity and mortality in these immune deficiency patients. Some of them developed autoimmunity and inflammation, but really it was the infections that were the big problems. But about 15 now, maybe close to 20 years ago, it was recognized that there were genes that caused immune system problems where the infections weren't such a big problem, but what happened is that patients developed severe autoimmunity or autoinflammation, and so these are defects in the regulatory part of the immune system. Again, the brakes that I told you get applied when the immune system gets gets activated, anything that causes either the immune system to get hyperactivated, so like, maybe the accelerator gets stuck, or maybe the brakes don't work, or that resolution phase doesn't work. These are the sorts of genes that fall in here. And if you take all of the 506, 511 genes, 560 defined clinical entities, but 511 genes, some of the gene defects are, some of the defects, the disorders are caused by defects in the same gene, but in one case, it's a loss of function of that gene. In other cases, it's a gain of function, and they cause two very different clinical syndromes. And so that's why there's 560 syndromes, but only 511 genes. Sorry. Excuse me. So if you take those and you divide them up between these primary immune deficiency diseases and what we call PIRDs, or primary immune regulatory diseases, so these problems with immune dysregulation, they split out about 2/3 and 1/3. So about 2/3 of the gene cause problems with immune deficiency or defects in responding to infections or insults, and about 1/3 of them cause dysregulated immunity. Okay. So that's what we're working on. So now let me just give you a clinical case, just as an example of what we see with these cases. So this is a real case, 4-month-old male. He presents to an outside ER in respiratory distress, and he has a chest X-ray that shows patchy infiltrates. And his parents say that he's had 3 episodes of ear infections since he was 2 months of age, which is pretty young to start ear infections, and especially that many. He had a 2-month history of respiratory dwindles. Basically, they said he had a cough, he was sort of snotty, and he was hospitalized once 3 weeks ago. It just seems to like it's getting worse and worse. He was hospitalized once 3 weeks ago. He got nebs and oral steroids, and he was modestly improved, so he was sent back home, but he's just not growing very well. And he's got his responses to the antibiotic therapy were just not very good for his lung infection. And so they brought him back in, and his bronchoscopy now shows pneumocystis jirovecii, which is, we all have it. It's in all of our airways. It doesn't cause you problems unless your T cells don't work. And now this is it used to be PCP pneumonia, now it's PJP pneumonia, and is one of the hallmark infections, for instance, of HIV. Once your T cell numbers drop below a certain level, you are susceptible to this. And so they did a bronchoscopy, and they found that in him, and his CBC showed lymphopenia, but it was so his T cells were very, very low. His B cells were normal, actually, but his NK cells were low. So you would say, boy, this looks like SCID with a T negative, B positive, NK negative phenotype. And based on that table that I just showed you, we can predict which gene it's probably going to be in this patient. Okay, so that's sort of a classic case. This is how SCID always used to present. Okay, but now we have SCID newborn screening, as I mentioned, and this picks kids up. And so this is from a paper that was published by Jennifer Puck, looking at the first 11 states that were screening, the first 3 million infants that were screened, and looking at what sorts of gene defects were found in those patients. And so the IL-2RG was the most common, followed by RAG and IL-7 receptor, but there's still a fair number where we don't find a gene defect, which is kind of interesting. About 20% of these patients don't have a gene defect, but they go on to receive therapy, and that therapy is either, most of them get a bone marrow transplant, okay, so like a pretty significant intervention. And many of the others who don't have problems with their, an intrinsic problem with their lymphocytes, their problem is with the thymus, where the T cells develop, the thymus. So in DeGeorge syndrome, patients um, with CHD7 mutations, things like that, that their thymus doesn't develop. They don't develop T cells either, because that's where the T cells need to go to develop. And so they have low T cell counts. And so they get a thymus transplant now. So bone marrow transplant or thymus transplant, either way, it's sort of like a big deal. So now you've got a baby that's born. They look totally normal because they haven't gotten all of those infections yet. You call them back to clinic, and now you're telling the parents, your baby has a life-threatening disease. And if we don't do this really aggressive therapy, they're going to die. And they're sitting there, the parents are sitting there looking at their baby saying, but they look completely healthy. They look great. And we say, well, they do now, but they won't stay looking great forever. In fact, probably for not very long. And you can imagine sort of the impact that this has and the challenge. Now, why does this matter? Well, these are from some studies that were done in the Primary Immune Deficiency Treatment Consortium, where we looked at bone marrow transplant before and after newborn screening. Actually, I'm going to jump to this one. So if you prior to newborn screening, this is a survival curve. Prior to newborn screening, the outcomes of, because patients came into transplant infected, survival was only about 80% of which is good, but not, I mean, it's not great. So 80% survival, this is what we tell families. And so they're thinking there's a 1 in 5 chance that my baby's going to die during transplant, which is what it was. So that was a problem. And when we looked at them, the difference was really that this group that did poorly were the ones that came in with infections. The group that did fine or did better were the ones who didn't have infections. So the whole idea of SCID newborn screening was that now let's pick them up at birth before they get infections, and will that improve outcomes of transplant in the end? And in fact, it does. And so transplant outcomes are markedly better in those patients that were identified by SCID newborn screening here in green than they are in patients who were identified in other ways. And so earlier diagnosis prior to infections and transplant early before they get infections improves outcomes to over 90%, 95% in these patients. So still not 100%, but pretty darn good. And so that's great for patients. It's great for families. The other thing that we have seen there is that disparities based on race and ethnicity, which there will be a paper coming out soon, documented prior to newborn screening, there were differences in outcomes between Caucasian infants and non-Caucasian infants. And what's interesting is that SCID newborn screening has erased that difference in outcomes because patients get detected universally early and get routed to an immunology center and the bone marrow transplant center where they can be treated. So it's pretty remarkable the benefits of newborn screening for this disease. So with that in mind, what are the most urgent needs for the work that ClinGen's doing, associating gene to disease and variant to disease? Well, these are among all of those IUIS genes that I mentioned, those 511 genes, these are the ones that require early, fast intervention with a really aggressive therapy, bone marrow transplant or thymic transplant. So it's really pressing that we clear up ClinVar and make it really clear which variants are pathogenic and which ones are not pathogenic. Because clinicians all over the country now, every state is doing SCID newborn screening. They get the genetics back and they go, is this a VUS or is this pathogenic? And yes, you can do other studies to figure that out in the lab, but those take time. And we clearly showed in this study from PIDTC that outcomes are better the earlier you can transplant decreasing the risk for infection. So that's why the work that you all are doing is so urgent. Okay, and so this is the SCID, CID, GCEP, and VCEP. We are funded by an R24 to try and get this. We've had R24 funding; it's just ending, and we're applying to be able to complete this work. But we have focused on tables 1 and tables 2, the SCID, severe combined immune deficiency, and combined immune deficiency tables of the IUAS gene list. That's our focus. Those tables had already been built. We figured there wasn't a reason to go back and try and rebuild that infrastructure. We'll just go with that. And so our GCEP is focused on that. These are the curations that we've completed and have been posted. And these are the ones that have been the curations have been done by other groups, and we just need to review those and related to the immune deficiency piece, and then we can move those over. And then we've got a few more to do, and then we'll move them over. We've begun to write the table 1 publication, and the table 2 publication will come shortly thereafter. And as you can see, most of these have been associated. These phenotypes are so strong that most of these have been associated with a definitive classification. There's enough cases to get them to definitive. There's a lot of strongs, but there are some moderates and there are some limiteds because we just don't have enough cases. Some of these are extraordinarily rare. The VCEP is focusing on the top 7 genes that have come out of SCID newborn screening that we know are the top hits that come out of newborn screening, so that we can clean up those and make those associations first, so that we can really make for providers who are needing to make these really time sensitive decisions, we can have that cleaned up and available for them. So that's where we're at. That's what we're doing. I just want to give you a sense of the importance of the work that you all are doing, and how it has real world impact for these patients and families. So with that, I'll stop. Happy to take any questions.
Thanks so much, Troy. And I at least can speak on behalf of myself, who doesn't live in the immune field, but that was an excellent overview of the immune system to make it approachable to all of us, I think. So really appreciate that. There is one question in the chat from Dimitri. Can defects in MHC1 or MHC2 cause SCID-like phenotypes?
They do. And so MHC, so again, those are, I think of them as sort of like a hot dog bun on a stick. Remember, those are the molecules that sort of present pieces of proteins from inside the cell. They present them to the T cells outside the cell. So you've got a hot dog bun with a piece of the protein in it from inside your cell. It might be a piece of the viral protein, and it's sitting on the end of a stick that's sort of the transmembrane portion of that. And the T cell receptor sees that. Now, the CD8 T cells see things that are in the MHC class 1, and the CD4 T cells see things that are in the MHC class 2. So you can imagine that a defect, so defects in the MHC molecules have been called bare lymphocyte syndrome. Basically, they basically, your cells can't, they're bare. They don't have, they're not able to show things from inside themselves. So, but basically, you can imagine that if there's a defect in the class 1 MHCs, then since it's the CD8 molecules that need to see that, and they need CD8s as they're developing, they need some stimulation through their T cell receptor to go on and develop. If there's no MHC to give them that stimulation, then CD8 T cells just never develop. And so MHC class 1 defects only lack primarily CD8 T cells. MHC class 2 defects only lack primarily the CD4 T cells. So again, as I mentioned, most defects lack all T cells. These are a couple that are sort of unique in that they only lack CD4s or CD8s. And as part of that, one of the challenges with that is that when you do that TREC testing for the SCID newborn screening, they've got some T cells. And so sometimes they'll come through with low Trex, but not low enough to meet the threshold for the newborn screening program. So this is among just a couple of genetic disorders, genetic SCID disorders that we see that can actually be missed by SCID newborn screening and can show up in the classic way with infections now a few months later. So hope that makes sense.
Yeah, that's very interesting. Have you guys discussed, is there any way, especially that transplant data, they're so compelling, is there any way to try and pick those individuals up? Like, if, I guess clearly there's a balance between picking up too many people and, missing a couple of those really rare edge cases. But it just
Yeah.
seems like those babies, it's so critical to catch them before infections.
It is, and there's been a lot of thought about how to go about that. There's not a great solution yet,
Hmm.
and so, and it's only a portion of them
Uh-huh.
that don't get picked up by newborn screening. So it's not all of them. So it that there's not a good there hasn't been a good solution for that yet.
Yeah, makes sense. Okay, let's see. Can you explain something more related to the different phenotypes associated to single gene variation when related to dosage sensitivity, haploinsufficiency, or triplo sensitivity, if a disease mechanism is due to copy number variants, so whole gene
Mm.
deletions or duplications, versus single nucleotide variants?
Yeah. So um, it's a great question, and it's one that we you know that we struggle with. What we've done in terms of the detection piece of this is that, for that, as I showed you, there's a subset of even patients who are picked up by newborn screening who don't have a genetic defect. Even when you do whole genome, you can't find anything. And where copy number variants testing, copy number variant testing is done as sort of a second tier in those. So that's one way to detect them. In terms of the dosage, there are absolutely some of these defects where dosage makes a difference. And so, um, one of the defects that can cause a thymic defect is FOXN1 deficiency. And there seems to be a clear association with the amount of gene function. So, um, you know, uh, as you get less and less function, um, you get less and less thymus essentially coming through. And so at a certain threshold, you get like really very little to no thymus. So there are defects where gene dosage is important. And so that can be important. It's important both from the standpoint of copy number variants, but it's also important from the standpoint of gain or loss of function variants, where they're missense variants that you might not lose 100% of function. So think about like the rag. So the rag genes. Remember those scissors that cut the loops out? So there's a whole variety of mutations in rag genes. So you can have a CNV that basically takes out the whole gene. Gone. That's severe. You have severe combined immune deficiency. No T cells, no B cells. Or you can have a point mutation in those genes so that now the scissors are there. They're just kind of dull. And so now they don't chop very efficiently. And so but every now and then they'll actually make a cut. And when they make the cut, that T cell gets past that checkpoint and now goes on to develop. And so now some of those T cells and B cells will leak through. And so we call those patients leaky SCID patients.
Mhm.
And so a few of those T cells and B cells will leak through. But now it's like putting a single dandelion seed in the middle of a very fertile field. Pretty soon you're going to have a whole bunch of dandelions in your field, but they will have all originated from that original
Mhm.
seed. That's what we see in patients with these leaky forms of SCID. They've got a ton of T cells, but they're oligoclonal. They all originated from the same T cell, or same limited number of T cells. And what happens there is that those T cells, as they divide, those TREX don't divide with them. They don't the TREX don't replicate, but so they get diluted out over time. So even though those individuals, those babies will have a lot of T cells, their TREC test will still be low because they have very few Tregs, even though they got normal T cells. So those still get picked up by the Treg test. So, but there, it all has to do with the level of functionality of those gene products, and it makes a difference. So,
That's so interesting. Well, we are at time, and so I just want to
okay,
reiterate again, we're so grateful that you came, and that was a wonderful overview and update of the
okay,
progress. So thank you
perfect,
again, and we will talk to
absolutely,
everyone in a couple weeks. Thank you.
awesome. Thanks, everybody.